So a kinetochore is the protein structure on chromatids where the spindle fiber is attached during cell division to pull sister chromatids apart. To drastically oversimplify what they look like, we'll do it this way. So this, that looks terrible. This is our chromosome composed of two sister chromatids. This is our centromere. I'll write bigger. This is our centromere. That is a region of DNA. The kinetochore is a separate structure composed of proteins found alongside the centromere, to which spindle fibers attach. So the kinetochore and the spindle fibers. So, the centromere is a region of DNA. The kinetochore is a protein structure, and the spindle fibers attach to the kinetochore. They do not attach to the centromere. Stating that is wrong. So, stating that the spindle fibers attach to the centromere is incorrect. Stating the spindle fibers attached to the kinetochore is correct. And the kinetochore is adjacent to or attached to the centromere is close enough. So mitotic spindles, which I call spindle fibers, because I call them that because they're easier to say that way. It's fewer syllables for me, and I like fewer syllables. They make me happy. The macromolecular machine that segregates chromosomes to two daughter cells during mitosis. Those are the spindle fibers. They are the things that are pulling those sister chromatids apart into two separate, identical chromosomes. A cleavage furrow is the indentation of the cell's surface that begins the progression of cleavage, by which animals and some algal cells undergo cytokinesis. Algal cells meaning cells that are algae. So a cleavage furrow looks like this. Insert jokes here. So, oh hey look, a scale bar. And if you're not immediately reaching for your ruler, I have one right here for you. Alright. So, effectively the cleavage furrow is formed by the function of a contractile ring, which is a ring of actin. It functions similar to a purse string. If you pull on that string, that will cause the edges to cinch together. Eventually, that cinching will continue until they separate into two different cells of roughly equal size, depending on what you need. So those are the two daughter cells that you get. The function of the movement of the chromosomes to either pole has already put the forming nucleus, the forming nuclear division into this and had made them separate, genetically identical daughter cells by this point. Now we have equal size daughter cells through the action of the cleavage furrow formed by the contractile ring during cytokinesis. Now, the question is, are plants different? And the answer is not that much. So, with plants, our prophase will still have condensing chromosomes. Our nucleolus will be present. And as prophase continues to later prophase, our chromosomes will continue condensing, and our nucleolus will get smaller and less obvious. Our nuclear membrane will degrade and become less visible. And then we'll switch to metaphase, where we will have the chromosomes lined up at the equator. Here we have spindle fibers that are kinetochore and non-kinetochore fibers. But what we don't have is we don't have the centriole. We don't have that actual organelle that is the complex arrangement of microtubules. But we still have spindles forming. We still have K fibers and non-K fibers, those kinetochore and non-kinetochore microtubules, which I'm calling K fibers and non-K fibers because those are also acceptable terminologies. We also have those fibers attaching or not attaching as required. In anaphase, we'll have the separation of these sister chromatids into individual chromosomes and their migration toward opposite poles of the cell. But the difference is not so much in telophase, where we'll see the decondensing of chromosomes, the reformation of the nucleolus, and the reformation of the nucleolus, and the reformation of the nuclear envelope. But here, where we see a cell plate forming, cytokinesis is what's different. Cytokinesis does not occur in plants. In plants, we instead have cellular division by means of the formation of a cell plate. So, a cell plate will form instead of cleavage occurring. That formation of the cell plate is the result of the Golgi-derived and endosomal-derived vesicles carrying cell wall and cell membrane components to the plane of cellular division. And they will fuse together, forming a new membrane and a new cell wall, a new middle lamella. And then, as that plate has occurred, it will fuse with the old cell wall, and that will cause separation of the cell, and new cell wall parts will be laid down, and the cell wall will expand from there, and the cells themselves will increase in size respectively from that point. Now, in this transmission electron micrograph image, not scanning, excuse me, we see our chromosomes, and we see the beginnings of our cell plate. We see the endosomal-derived vesicles forming along this equator right here. We should avoid calling it the middle of the cell, because this cell is not an even shape. This has more of a pentagon shape, so how do you find the middle of the pentagon? And it's not being split down this way. It's being split down this way. So this is not quite the middle, it's the equator. We'll call it the equator. Or this is just our cell plate, and we'll just leave it being called the cell plate. So here's our difference. We have in our animal cells and our plant cells, we have a microfilamentous ring of actin drawing in and forming a cleavage furrow until we separate into two separate cells. And in plant cells, we have the Golgi and the endoplasmic reticulum depositing vesicles of the new membrane forming a cell plate and a middle lamella. That middle lamella will thicken as more and more cell wall components are embedded into that middle lamella, thickening it into a new cell wall component. And we'll have two identical daughter cells formed by a cell plate. The new formation of a plasma membrane, two plasma membranes and a middle lamella, expansion of the middle lamella into a cell wall. Now, we've already discussed what the spindle fibers are doing during mitosis. Just as a quick recap, they are separating and They're separating, the centromeres are separating, going toward opposite poles. We have our kinetochore fibers and our non-kinetochore fibers attaching. We have metaphase, then we have anaphase where we separate them. And then we have where we separate the chromatids into chromosomes. And then we have telophase where we have our nucleus reforming. And that's pretty much it. That's what's happening for the spindle fibers. And we've seen this already in a textbook format. So in our other format of the video. Now, let's talk about genes for a moment, and not the kind you wear, the kind you are. So, genes are a sequence of DNA monomers that codes for a product. It is a sequence of DNA base pairs. So the product is different. The product is a different macromolecule. It requires a specific sequence of monomers, usually a polypeptide. However, the products could be RNA or tRNA as well. mRNA is just an intermediary molecule that serves the translation of the end products of protein primary structure. So, typically, what we're going to define DNA and what we're going to define genes as is a sequence of nucleotides on a region of the chromosome that codes for a polypeptide. That's going to be our working definition for AS level biology. It's going to be our working definition of a gene. That's specific phrasing, and you should probably go back and listen to that again. Now, let's talk about mutations. A mutation is a change to the sequence of nucleotides that may or may not result in an altered polypeptide. So, what is primary structure? I'll give you a pause to actually answer that question. Or you can go back to a previous video and go look it up. I don't have to answer that. That's what YouTube's for. Secondary structure. What's that? I will say this. Secondary structure is determined by hydrogen bonding. And that hydrogen bonding pattern is determined by the primary structure. Tertiary structure. What is that? What determines tertiary structure? What bonds comprise tertiary structure? And how did we get to that tertiary structure? Tertiary structure is built upon secondary structure, which is built upon primary structure. And tertiary structure determines what? It determines the 3D conformation of a single polypeptide chain. The 3D conformation of a single polypeptide chain. And I will leave that to the internet to remix and auto-tune as required. Now, what does, and that 3D conformation is going to determine the function of that protein. Structure determines function, and 3D structure is structure. So if structure determines function, tertiary structure determines the function. If tertiary structure is determined by secondary and primary structure, that means that ultimately primary structure determines function. If you go far enough back, if you chicken and egg this far enough back, then primary structure is very important for your function. And if you change something, if you alter that polypeptide and you alter that tertiary structure, you will change the function of that protein. So that brings us to the idea of alleles. Alleles are genes that are responsible for polypeptide sequence. Genes are responsible for a polypeptide sequence, which determines the function of that polypeptide. The function of those polypeptides are responsible for traits, features, or characteristics of an organism. If there is a gene mutation in that gene that results in an alternative form of that gene, it doesn't have to still be functional, that gene. It could still be functional. It doesn't have to be. That's what a mutation does. Alternative versions of genes, whether they function or not, or whether they function differently or not, those are alleles. So alleles are different versions of a gene. Different versions of a gene are alleles. So inheritance isn't discussed until A-level biology, and because these are AS videos, I'm going to leave this here. And if I ever need to make a series of videos on A-level, we'll come back to this. but not today. So DNA is in the nucleus for a couple of reasons. For one reason, it's big. For reason two, it's valuable. For reason three, cytoplasm is a dangerous place. Stuff tends to get digested out there, and it'd be a shame if something was to happen to you. So DNA has a job to do. It's an important job, but DNA is trapped in a gilded cage. A gilded cage that is the nucleus. That gilded cage, it needs to be able to get messages out to the cytoplasm where the stuff is actually happening, where the real action is, to the big city. Sorry to all the rural folks watching this. So second messengers is what it needs to send messengers for that. So what messenger will DNA send? If you said messenger RNA, you were right. So this is the central dogma of biology. The central dogma is that DNA undergoes transcription to form RNA. RNA undergoes translation to form protein. That is the central dogma. DNA transcribed to RNA, RNA translated to protein. That's the central dogma. The central dogma of biology. Now, the actual phrase central dogma, the way it's defined, sounds like it's from the 70s or 80s, because it is. And can you spot the reason why I think that, or know that for a fact? The coded genetic information hardwired into DNA is transcribed into individual transportable cassettes, composed of messenger RNA, mRNA. Each mRNA cassette contains the program for synthesis of a particular protein, or a small number of proteins. So, I wonder why I think this is from the 70s and 80s. Alright, so, now we're going to discuss how the code is transcribed and translated. We discussed somewhat briefly this when we talked about RNA before, but now we're going to talk about it in much more detail. The transcription of RNA from DNA is extremely similar to the process of DNA replication, with the replication fork, the separation of DNA, the placement in the 5' to 3' direction of nucleotides, the formation of phosphodiester bonds, but effectively, here's our similarities. The protein that transcribes RNA is RNA polymerase. It behaves like DNA because it lays down complementary nucleotides continuously, anti-parallel, in a 5' to 3' direction only. This is all taking place inside the nucleus. And while it's also happening during S phase, it's also happening during G0, 1, and G2 phase, of course, as well. So that's a difference. So both of these will happen during interphase, and that's your similarity here. The difference being that DNA replication is occurring during S phase, and RNA transcription is occurring during S phase, G0, 1, and 2. Now, more differences. Obviously, RNA is the final product instead of a DNA copy, an RNA transcript instead of a DNA copy. So the 4 letters of code being A, G, C, and U for RNA. RNA polymerase does not require a primer because DNA required an RNA primer. RNA polymerase does not require an RNA primer because what's going to make an RNA primer for RNA polymerase? RNA polymerase? What's it going to attach to? Mystical RNA? Another mRNA?